Welcome to our presentation on the neurohumoral transmission in the autonomic nervous system. What do we mean by neurohumoral transmission? So when we say neurohumoral transmission, that is the process by which the information is communicated or transmitted from nerve to nerve and to effector organs. So this involves the release from a nerve or a neuron terminal of a chemical neurotransmitter that reacts with the receptors on the innervated cell. So what do we mean by a neurotransmitter? So a neurotransmitter is a chemical substance within the synaptic vesicle that mediates the information transfer between the nerve cells at synapses. So basically when we say synaptic vesicle, that is a structure that is found at the axon terminal. So this diagram here shows a synapse, and when we say synapse, that is a junction with a minute gap that separates two neurons or nerve cells. They are also found between a neuron and a muscle cell or a gland. It is also defined as a specialized site of apposition where information passes from one nerve cell to another cell. So this diagram here shows a synapse, and this part here is the axon terminal. So this is the presynaptic neuron, and this is the postsynaptic neurons. Of course, this is the axon terminal, and this part here are the dendrites of the another axon. But of course, this postsynaptic neuron here in other synapse, it could also be another postsynaptic cell, and that could be a muscle or a gland. As you can see here within the presynaptic neuron, particularly here at the axon terminal, we have here the presence of the synaptic vesicle, and the synaptic vesicle contains the neurotransmitter. And the gap here between the presynaptic neuron and the postsynaptic neuron is referred to as the synaptic cleft. So this is another diagram showing the location where the neurotransmitters are located. We have here the synaptic vesicle. It contains within it the neurotransmitter. So this is the presynaptic neuron, and this is the postsynaptic neuron. This is the synaptic cleft. So the part of the neuron that contains the neurotransmitter are known as the axon terminal. So we also have here the presence of the voltage-gated calcium ion channels. And in the postsynaptic neuron, we also have the presence of the uh, receptors of the neurotransmitters. So the synapses, so of course this is the synapse. Now the synapses are composed therefore of the presynaptic neuron, the synaptic cleft, and the postsynaptic neuron. So the part of the presynaptic neuron that is shown here is this axon terminal, and the part of the postsynaptic neuron that is shown here is the dendrite spine. The presynaptic neuron again contains the terminal button, having neurotransmitters within the synaptic vesicles. The postsynaptic neuron is composed of a part of a dendrite or a cell body of the neuron and contains receptor sites to receive incoming signals from the presynaptic cell. The synaptic cleft is defined as the gap between the presynaptic and the postsynaptic neurons. So the autonomic nervous system is functionally divided according to major neurotransmitters that it releases. So the first neurotransmitter is the norepinephrine or the NE, also known as noradrenaline. So again, this is the major neurotransmitter at the terminals of the postganglionic fibers of the sympathetic neuroeffector junctions. Another neurotransmitter under the sympathetic nervous system is the epinephrine or adrenaline. It is released from the chromaffin cells in the adrenal medulla. We also have another sympathetic neuroeffector junction, and that is on the sweat glands in humans. This is an exception because it releases the neurotransmitter acetylcholine. So another very important neurotransmitter is acetylcholine or the ACH. It is the neurotransmitter at all ganglia, meaning for both parasympathetic and sympathetic, and at most parasympathetic neuroeffector junctions. So of course, we can use the neurotransmitter to classify the nerves, according to their function. So those nerves that release acetylcholine are known as cholinergic nerves. So it can be parasympathetic postganglionic, parasympathetic or sympathetic preganglionic, and in few regions asympathetic postganglionic nerve. So an example of this sympathetic postganglionic nerve is the um, neuroeffector junction at the sweat glands. No, sweat glands. So of course, for the parasympathetic, we have acetylcholine as its major neurotransmitter. So of course, here are the summary of the different neurotransmitters that are being released in the sympathetic and the parasympathetic outflow. So this portion here represents the scene as the preganglionic axon, the ganglion, postganglionic axon, and the effector cell. So between the postganglionic axon and the effector cell is the neuroeffector junction. When we say neuroeffector junction, that is the junction between these two structures. Of course, effector cell is also known as the target tissue. So in the preganglionic axon of the parasympathetic nervous system and the preganglionic axon of the sympathetic outflow, it releases both acetylcholine. So both of these are cholinergic nerves. For the parasympathetic postganglionic axon, it releases acetylcholine. So it is considered to be cholinergic in nature. We also have those that releases norepinephrine at the neuroeffector junction. So those nerves are known as adrenergic nerves. And this is under the postganglionic axon of the sympathetic outflow. These are the physiological events in the neurohumoral transmission, or the transmission of the signals or the impulse from one nerve to another nerve. So of course we have here the first is the axonal conduction. It is followed by the release of the neurotransmitter, then the receptor events, and the catabolism of the neurotransmitter. So the first is the axonal conduction. That is the first step in the neurohumoral transmission. So when we say axonal conduction, it refers to the passage of the impulse along a nerve fiber. So the impulse that we refer here is, of course, the action potential. It is dependent upon the changes in the permeability of the axonal membrane to the electrolytes. So this means that this portion of the axon is dependent on the permeability to electrolytes. Example are the calcium, sodium, potassium, and chloride. So of course, this important to induce later on the release of the neurotransmitters in the synapse. So for this diagram, you have here the presynaptic neuron, the synapse, and the postsynaptic neuron. So the part of the ending part or the end part of the axon that contains the synaptic vesicle is known as the axon terminal. And each axon terminal is swollen, forming a synaptic knob. The synaptic knob is filled with a membrane filled with membrane enclosed vesicles, and that is known as the synaptic vesicle. So those structures that are within the synaptic vesicle are neurotransmitters. So depending on the nerve, so this could either be acetylcholine or norepinephrine. And of course, we also have here the presence of the voltage-gated calcium channel. And in the postsynaptic neuron, we also have here the presence of the receptors for the neurotransmitter. So later on, we'll be able to identify what are the functions of this voltage-gated ion channel and what are its importance, as well as the receptors that are found in the postsynaptic neuron. The second step in the neurohumoral transmission is the release of the neurotransmitter. So this is triggered by the arrival of the axonal action potential at the nerve terminal. So one of the important mechanism that triggers the release of the neurotransmitters is the calcium ions. So the calcium ions act to link or couple the excitation of the membrane with the discharge of the neurotransmitter from the axon terminal. So again, this is a schematic diagram of our axon terminal. We have here our synapse, synaptic cleft, and the effector cell that contains the receptors. So within the axon terminal or the synaptic knob is this synaptic vesicle or the storage vesicles that contains the calcium ions. So when the action potential reaches the axon terminal, it changes the permeability of the axon terminal, allowing the entry of the sodium ions and the efflux of the potassium ions. So when the sodium ions will enter the cell, it will induce the depolarization of the cell. So this action potential will also, that will produce also the action potential. The action potential will also induce the opening of the voltage gated calcium ion channels. So the electrochemical gradient will be calcium ions will enter the cell, and this again will trigger the excitation of the membrane so that the synaptic vesicle will be triggered to move towards the end part of the axon terminal, and the synaptic vesicle will fuse with the protein that is present in the axon terminal, and it will induce the release of the neurotransmitters towards the synaptic cleft. So the neurotransmitters are released in the synapse, as shown in the figure. The nerve impulse at the nerve terminal triggers the release of the stored neurotransmitter, which may be NE or ACH depending on the type of the nerve. So this can be the storage vesicles could contain, for example, norepinephrine or acetylcholine depending on the type of the nerve, whether it is cholinergic or adrenergic nerve. So again, the important stimuli here is the arrival of the action potential and the entry of the calcium ions in the cell in the action terminal, allowing the storage vesicles to dock onto the action terminal to release its neurotransmitters by way of the process of exocytosis. So after the release of the neurotransmitters from the synaptic vesicle into the synapse, so of course, some of those neurotransmitters will bind to the receptors that are found at the postsynaptic cell, postsynaptic cell body, dendrite, or the effector organ. And these are known as the receptor events that take place in the neurohumoral transmission. So at this part, the neurotransmitters will bind to the receptors on the postsynaptic membrane. And these receptors are considered to be ligand-gated ion channels. So the postsynaptic cell contains the cell surface receptors. So binding of the neurotransmitter to these receptors will elicit a response. And when we say postsynaptic receptors, these are it is where the neurotransmitters would act when secreted into the synaptic cleft. So these are again ligand-gated ion channels, meaning that when the neurotransmitters will bind to these receptors here, it will open the receptors, and when the neurotransmitter is stimulatory, it will induce the opening of the receptors, opening of the ligand-gated ion channel, allowing the entry of calcium ions into the cell so that the action potential will be triggered in that cell. So when the neurotransmitter is inhibitory in nature, it will induce the hyperpolarization of the postsynaptic cell. So meaning that it will promote the entry of chloride ion, which is a negative ion, and the exit or the efflux of potassium ions out of the cell, no? Triggering hyperpolarization and inhibiting the action potential. So after the neurotransmitter is bound to the receptor, so this would trigger, the opening of the ligand-gated ion channels, in the postsynaptic cell. So within the postsynaptic cell, there are different types of voltage-gated channels. We have the sodium gated channel. We also have the potassium voltage-gated channel. We have the sodium and potassium leak channels, now that maintains the membrane potential, and we also have the calcium, calcium ion channels. So the binding of the neurotransmitter to these receptors again would open the ligand-gated sodium channels and generate impulse in the post-junctional fibers. Of course, when the sodium ions will enter the cell, now that will trigger the generation of the impulse through the action potential. It would cause a response which is terminated when the NT dissociates from the receptor. So there are two types of receptor events. The neurotransmitter could either be induced excitation or the neurotransmitter could induce inhibition. So of course, there are certain types of neurotransmitters that are excitatory in nature, while there are also those types that are inhibitory. So when we say excitatory response, that happens when the neurotransmitter will increase the electrical excitability on the postsynaptic side through modulation of the transmembrane ion flow to facilitate transmission of the action potential. So basically, pag excitatory, it involves the excitatory neurotransmitters. Now, and this only means that the neurotransmitters will excite the neuron and cause the firing off of the message. So meaning that the information is transmitted from one cell to another in the effector organ, for example. And the message continues to be passed along the next cell. So that is an excitatory response, and that is induced by excitatory neurotransmitters. Now, for example, the norepinephrine. No, norepinephrine is an example of an excitatory neurotransmitter. In the ANS, we also have the epinephrine and the acetylcholine. So what they do is they will depolarize the cell to trigger, the generation of the action potential. And that action potential is important for the production of the response to that particular cell. So what do we mean by inhibitory response? So when we say inhibitory response, of course, this involves the inhibitory neurotransmitters. So these inhibitory neurotransmitters will block or prevent the chemical message from being passed along the nerve fiber towards the another cell. So of course, this involves the inhibitory neurotransmitters. So these neurotransmitters will hyperpolarize the postsynaptic membrane. When we say hyperpolarize, it will increase the negativity of that particular membrane, inhibiting the generation of the action potential. So what it does, for example, is that it will allow the chloride ions, which are negatively charged, to enter the cell and induce the efflux or the movement of the potassium ions outside the cell. Example of this is the gamma aminobutyric acid or the GABA. This is considered to be uh, the GABA is considered to be a major neurotransmitter in the central nervous system. And it induces an inhibitory response. This diagram shows the representation of the excitatory and inhibitory synapses. So there are two major ions that are important for the the manifestation of the excitatory and the inhibitory response. And these are the sodium ions and the chloride ions. So their movement in and out of the cell is important for depolarization and hyperpolarization. So in an excitatory synapse, so we have here the axon terminal, the synapse, and the postsynaptic membrane. So glutamate is an example of an excitatory neurotransmitter. When it will be released, of course, this is in the central nervous system. When it will be released in the synapse, what it will do is it will bind to its receptor in the postsynaptic membrane. And what it does is it will open, now for example, the sodium gated ion channels, and it will allow the entry of the sodium ions into the cell, inducing the depolarization of the cell. So when we say depolarization, that is an increase in the reduction in the negativity of the cell. So magiging positive yung cell until it reaches the, this is important, no, to induce the action potential and to trigger the to promote, no, the movement of the impulse, no, from one cell to another, and ultimately to produce the response. For an inhibitory synapse, it will, for example, it will example of this is the GABA aminobutyric acid, which is a major inhibitory neurotransmitter in the CNS. So what it does is it will allow the entry of the chloride ions, which is the negative ion, towards the cell of the postsynaptic membrane. So since this chloride ion is negatively charged, so it will increase the negativity of the membrane of the cell, thus inhibiting the generation of the action potential. So when there is no action potential, of course, there will be no response that will be initiated. So not all of the neurotransmitters will be able to bound to the receptor. Some of those will be broken down in the synapse, and some of those will be reuptake, will undergo reuptake towards the presynaptic cell or presynaptic neuron. And this process is known as the catabolism of the neurotransmitter. So in the case of the NE or the norepinephrine, uh, before that, no, we have first to define catabolism. So when we say catabolism, that is defined as the chemical or enzymatic reactions involved in the breakdown of neurotransmitters. So basically, it terminates the duration of action of the neurotransmitter. So in the case of the norepinephrine is again the neurotransmitter at the postganglionic neuron of the sympathetic nervous system. So it will, some of the norepinephrine in the synapse will undergo reuptake into the presynaptic terminal via the NET or the norepinephrine transporter. And then the transmitter is either degraded within the cell by the enzyme MAO, monoamine oxidase, or COMT, catechol O methyltransferase, or repackaged into the synaptic vesicles. This diagram shows the catabolism of norepinephrine. This is the axon terminal, this is the synapse, and this is the postsynaptic cell. So we have here in the axon terminal, we have the synaptic vesicle, and we have here a structure showing the synaptic vesicle undergoing exocytosis. So it releases neurotransmitters in the synapse now to act on the receptors that are located in the postsynaptic membrane. So not all of the receptors will be able to bind, and some of the neurotransmitters or the norepinephrine will be broken down, and some of those will undergo reuptake, into the presynaptic nerve. So how does the norepinephrine action terminated? So the action of the norepinephrine is terminated by reuptake into the presynaptic terminal via the NET or the norepinephrine transporter. So what it means is that some of these norepinephrine will undergo reuptake into the presynaptic membrane by way of this transporter. And when they reach the presynaptic terminal, they can either be degraded by MAO, monoamine oxidase, or catechol O methyltransferase, and others will undergo repackaging. So other neurotransmitters, norepinephrine, will undergo repackaging into the synaptic vesicle. So that is now the mechanism of the catabolism of the norepinephrine after it is released in the synapse. So in the case of the acetylcholine, the acetylcholine action will be terminated by acetylcholinesterase, which is an enzyme present in the synaptic cleft. The enzyme will degrade acetylcholine into choline and acetate molecules. The choline is then transported back into the presynaptic terminal and used in the synthesis of new acetylcholine. This diagram shows the catabolism of acetylcholine, which is the major neurotransmitter in cholinergic nerves. So this is again the presynaptic nerve terminal, or the axon terminal. We have here the axon synaptic vesicle containing the neurotransmitter acetylcholine, and we also have here the synaptic cleft and the postsynaptic membrane or postsynaptic cell. So this portion here represents the process whereby the acetylcholine is being released now from the synaptic terminal. We have here some of the acetylcholine will be able to bind to its receptors, and some will be broken down and will undergo catabolism. So acetylcholine is degraded into choline and acetate by the enzyme acetylcholinesterase. So in the synaptic cleft, you have the presence of the enzyme ACHE, which will break down acetylcholine into two parts or two components. We have the choline and the acetate. What will happen to the choline is that it will be transported back into the presynaptic terminal. So that is now the process whereby acetylcholine is catabolized after released into the synaptic cleft.